Namaskara Karnataka, I'm Miss Gopika, your own biology master teacher. Welcome all of you for another amazing session for your preparatory boards, right? So pre-boards are very important, as same as boards. Why? Because you'll know exactly what you're supposed to write, what kind of questions will come. But let me tell you, pre-boards are always going to be a little difficult than boards, okay? This you have to always keep in mind, but that does not mean you don't prepare at all, okay? It will be slightly difficult than boards, because boards usually the paper is in a range of easy and medium. It's never hard until the paper is leaked multiple times, okay? So there's always, multiple sets of paper that will be made. So usually you will get this easy to medium range of papers, but your pre-board papers would be usually a little or a slightly difficult one. So let's see some of the questions that are, that I have high chances to come for your this pre-board exam, okay? Hi Bhumi, I'm doing good. I hope all of you also are doing good. Please let me know in the chats how all of you are doing. It's been some time that we met, right? Yes, tell me students. Am I audible, clearly visible? There is no problem with the live, right? Let me also check. Confirmation. Yes. Perfect. Everything is clearly visible. Hello. Hi Saujanya, very nice name. I'm doing good. Okay. Yes. Shall we start? So 70 on 70 pakka madana ee time. Alva, biology. So we will do top 30 questions, okay, 25 to 30 questions. We will take some questions are repeating multiple times, so I have avoided that question, but I have taken these questions from the model question papers and from your previous year, all the question papers, wherever high repetition has happened, but I cannot include all of it, because if I include all of it, it will become a very long session and you will also be confused. My God, this ma'am has told whole chapter. So there are some questions that are, repeated, so that photos I will be sharing on the Telegram channel. So all of you run and go join We Karnataka, KCET and both's Telegram channel, and you can also join my Telegram channel, that is Biology by Gopika. Okay, yes, very nice. Okay, let's start the class, students. First question, okay, first question, I hope it's clearly visible. How does copper T act as a contraceptive? So you will neither get copper T, or they can ask you all the IUDs, because IUDs is one of the most common contraceptive methods that is used in India, and it cannot be inserted by us, like the people, normally people cannot insert it, just like how we insert condom, it cannot be done. We need a practitioner, we need a female nurse, female nurse who can do this for us, okay, contraceptives. But this question has come multiple times in different ways. They have asked what is IUDs, they have asked how IUDs protect. Now here also you can see how does copper T act as a contraceptive. That means they're asking you what is the function, how is copper T exactly going to help as a contraceptive, okay? Ready all of you for the answer? So release of copper ions is the first thing. This I have taken exactly from your NCERT. I have just made it in a better way for you to understand, okay? The copper T releases copper ions in the uterus. Now what is the thing? This is inserted into the uterus, okay, through the vagina. Now the thing is, they are going to release copper ions in the uterus. Now what this copper ions are exactly going to do? Sperm is going to come, right? When the sperm enters, they will make sure that the sperm will not be able to survive this environment. Now let's say I'm putting Bhumi and Saujanya in a desert and I'm telling that you people live here for 1 month. Obviously Saujanya and Bhumi will not be able to leave, because this is not the environment that they lived in, alva? So they will not be able to live. They'll be like, ma'am, aagta illa ma'am, tumba sake ma'am, okay? So it's too hot and ತುಂಬ ಶಕೆ ಇದೆ ಮ್ಯಾಮ್, ನೀರಿಲ್ಲ ಮ್ಯಾಮ್, ಫುಡ್ ಇಲ್ಲ ಮ್ಯಾಮ್, ನನಗೆ ಬಿರಿಯಾನಿ ಬೇಕು, ಏನು ಮಾಡೋದು? So they will ask me 100 questions and they'll tell they cannot survive. Same thing here also. When the sperm enters, they will make sure that it does not give an environment for the sperm to survive, okay? So toxic effect on the sperm. Copper ions have a toxic effect on the spermatozoa, reducing their motility. What is motility? That means their ability to move, right? They are moving all the way from there and coming towards us, right? Yes or no? That keeps varying. That is one problem, okay? Only boards will be, all of you will have the same dates, otherwise everything will be continuously changing, okay? And their viability, that means for how long these people will be active. In a female's body, a sperm can survive up to 4 to 5 days, okay? So they will make sure that the sperm's viability also reduces. This prevents the sperm from reaching and fertilizing the ovum. Clear to all of you? So the first thing that they do is they will release copper ion. Second thing they will do is they will make sure that they do not give a proper environment for the sperm to survive. Okay, now alternation of the uterine environment. Copper also alters the uterine environment, making it unfavorable for sperm penetration and implantation of the fertilized egg. Okay, they will always affect the implantation, and also they will make sure that the sperm does not feel happy, okay, all jingalala. It does not feel, it will be stressed. Okay, it will be stressed that, oh, you no viability, I can't move also, they have reduced my fertility. So all that kind of things this copper T will be doing. Okay, or normally your IUDs, be it copper T, be it multi-load 375, be it copper T, okay, all these people, or copper 7, all these people will be doing the similar kind of function. Okay, thumbs up in the chat if all of you understood. Thumbs up. Okay, give another name. Okay, I want all of you to like the video and share the video with your friends, like I always tell. Okay, 1 second. Yes. I can't see your chats. Oh, I'm so sorry. One second, students. Yeah, okay. Yes, all of you have to respond, okay? Don't sit silently. Don't be like, oh, what will ma'am think? I'm very shy. There's nothing to be shy, right? You just have to put a thumbs up. Now, give another name for sexually transmitted disease and name 2 sexually transmitted disease which are curable and 2 diseases which are not curable. So, they're talking about what? They're talking about STDs. Okay, STDs, that is sexually transmitted disease. Now they're asking us which are the diseases that are curable. Like if we get a sexually transmitted disease, which can be cured and which cannot be cured. Okay, so curable diseases are syphilis, okay, which is a causative agent, you don't have to know, but again I have just written it here. Treponema pallidum, which is a bacterium, which you don't have to remember because it's not there in your NCERT. Then you have gonorrhea. Again, Neisseria gonorrhea, again a bacterium. These are the causative agents. So these 2 are the curable ones, and the non-curable one. Okay, not curable one is first one. Obviously, all of you know it's going to be HIV and or AIDS. Okay, HIV/AIDS. HIV, AIDS, and second one, genital herpes. Okay, so these 2 will not be curable. That means there is definitely antiviral drugs will be given. Okay, antiviral drugs will be given, but the thing is, students, these can only be, you know, we can just maybe little prolong their life period, okay, the span, or we can, you know, reduce the reactions. Other than that, we cannot cure it completely. Okay, so these 2 will come under the not curable and these 2 will come under curable. This is a very simple question. They will ask you what is STDs, they can ask you what is AIDS, they can ask you what is HIV. So all of this are on the similar background. Okay, understand that on a general, the outline is going to be the same. Okay, outline is going to be the same. Now, how do the following contraceptives act to prevent unwanted pregnancies in human females? First one, they have given IUD. So in IUD, all of you write, first of all, see, it's already given intrauterine device. Whatever you wrote for copper T, no, you can write the similar thing, that they do not provide a proper environment for the sperm. Okay, they do not provide proper environment for sperm. They do not, okay, do not provide proper environment for the sperm. At the same time, they make sure that the uterine wall also, okay, uterine wall also okay, it does not allow implantation. So I'm just writing it in points. You can just write the same points which we just discussed. Basically, understand these people will be trying to do maximum things so that fertilization does not happen. Okay, is it clear to all of you? Yes. Tell me. Yes, you can also call it what? Sexually transmitted infection. You can call it reproductive tract infection. You can use those words, okay? Yes, Nishmita. Yes, now next one, students, Saheli. See, Saheli acts as a selective estrogen receptor modulator. That means what it is going to do? Saheli is one of the oral contraceptive. That means you can take it orally, which does not have maximum side effects, because most of the combination of your oral contraceptives, it is going to be in this combination where it is estrogen plus progesterone or progesterone alone. Okay, this is going to be the combination usually of your oral contraceptives. So Saheli is one such good medicine, okay, one such good tablets which is very affordable. First of all, dosage is very it's not high dosage that it causes extreme side effects. That kind of things is not there. That is the reason why Saheli is so famous and so spoken. Okay, so Saheli acts, what does it do? It blocks the effects of estrogen in the uterus while promoting its effect in other parts of the body. This prevents the implantation of a fertilized egg. I told you, everything that these people are going to do is going to affect the implantation. If you only affect the implantation, then we can do something about it, right? Or we have to slightly suppress the ovulation. Such kind of things usually happen. Now, inhibition of ovulation, in some cases, Saheli some cases only partially suppresses ovulation, further reducing the chances of pregnancy. Obviously, it's a contraceptive. The main job should be reducing the chances of pregnancy. Okay. Now, Saheli alters the endometrial lining of the uterus, making it unsuitable for implantation of fertilized eggs. So students, this point you can write it mainly, that Saheli mainly does the job of making the uterus unsuitable for implantation. Any doubts in this? All of you like the video? Yes, all of you smash the like button. Okay, so these are the things about Saheli. Now, again on this basis, as I told you, maximum questions have come from IUDs. Okay, I have seen maximum questions from IUDs itself. What is IUD? What is the full form of IUD? Give the different types of IUDs. Talk about non-mediated IUDs, hormonal IUDs. These are the general questions that have come. So, all of you can prepare for that. Like this Lippes loop is there, non-mediated IUD. Then your hormonal IUDs are there at the progesterone, okay, progesterone or LNG20 is there. Two examples you can remember. Or you have to remember about the copper Ts, that is copper releasing IUDs like copper T, copper 7, multi-load 375. Know the names of it. That is more than enough. If you know the names itself, you will get 1 mark for each of it. All these are 3 marks, 2 marks, 5 mark question. So all the names itself will give you half mark or 1 mark. Okay, half mark system is not there. So directly they'll go for 1 mark system. Now, describe beads on a string structure of a DNA. Here, what are they talking about? Ma'am, here they are talking about packaging. Here they are talking about packaging of DNA. I told you, if I give you a long thread and ask you to put that thread inside the nucleus, you will be like, ma'am, what is wrong with you? What are you telling? How can I do this? Right? So for that, whom did we take the help of? We took the help of some proteins which are called as histone proteins. Histone proteins are positively charged. DNA is negatively charged. So positive and negative will attract each other, right students? So this long DNA, okay, this 2.2 approximately 2.2 meter long DNA will have to enter into the nucleus. Yes ma'am. To make them enter into the nucleus, we have taken the help of histone proteins which are positively charged. Now those histone proteins are arranged in histone octamers. Octamer means what? Each of them have come as a couple. Okay, two people, like that. Four histone proteins have come as a couple. So it became octamer, 8. Okay, octamer. Now with that octamer, when DNA is wounding around, we will call it what students? We will call it a nucleosome. I'm teaching you the concept also because they have asked you questions about what is packaging of DNA. They have asked you question about what is nucleosome in 2 mark question. So everything you will be able to answer from this itself. Okay. So look here, histone octamer plus DNA together is called as nucleosome. Okay. Now when I view it under a microscope, that means in the field of the microscope, no sound. Is it only for Ali Khan? So look here. There's sound here. Please check Please check, switch on your Bluetooth. Okay, yes. So histone octamer H1 protein. Now students, to keep all of these people in place, we have one protein which is called as a linker protein, which is mentioned in your NCERT. So they can ask you as an MCQ. Okay, MCQ. That is H1 protein. What is H1 protein called as? It is called as a linker protein. Okay. Now, histone octamer plus DNA together, it's called as nucleosome. Okay, nucleosome. Now, this here, the most common amino acids, okay, most common amino acid is going to be what? Lysine and arginine, which is again mentioned in your NCERT. Now, what did I tell you? If you see it under the microscope, it will look like a bead on a string structure. It will look like what? Bead on a string structure. Clear or not? That is the question for you. Now, remember students, a lot of condensation will happen leading to chromatin fibers and then your chromosomes. Now, let's go back to the question to understand what is given in the question. Now, describe the bead on string structure of a DNA. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called as nucleosome. Done? Okay, perfect. Okay, a typical nucleosome contains 200 base pairs of DNA helix. What are they telling? Base pairs at Gopika ma'am's class. Okay, always remember this, at Gopika ma'am's class. Adenine will always bind with thymine, guanine will always bind with cytosine. Okay, so 200 base pairs are going to be there. Now, nucleosome constitute repeating unit of a structure in a nucleus called as your chromatin thread, like stained colored bodies. I taught you, euchromatin, heterochromatin. So these are going to be stained. Now, all of you have done root tip mitosis, meiosis experiment in your practicals. How many of you have done? Raise hands in the chat box. I have done. Okay, how many of you have done in your PUC? You have done onion root tip. I think, I don't know for others what they have given. Okay, so we have done mitosis and meiosis. So when you were doing mitosis and meiosis, all of you would have seen this one stage, no? In your, when your teacher is asking you to show the stage, most of you would have seen this, something like this. Okay, and you would have told, ma'am, I saw prophase. Then she'll tell, no, you should say something else. You should see at least anaphase, metaphase. So what you were seeing, students, they were basically your entire chromatin fibers, okay, which were not arranged properly. Later condensation will happen and slowly you will start seeing them in metaphase, anaphase, telophase, daughter cells will be formed, right? So that is what you saw. You saw a prophase, that was entirely your chromatin scattered. Now here you see the nucleosomes in the chromatin are seen as a bead on a string structure when viewed under the microscope. So in case this question comes for you, please write what is histone octamer, please write what is nucleosome, and please write how many base pairs will be present in one nucleosome. This will give you the full marks, okay? Because it's bead on a string structure is just a, like how I tell, oh, Taj Mahal is this beautiful, or I give a, I try to relate it with something, no, like that only. They have given bead on a string structure. Okay, that's it. Perfect. Moving forward, this is also clear. Now, explain the clover leaf structure of tRNA. Students, I have seen this as a very important question because many questions on similar basis I have seen. Like they have asked, explain tRNA structure. Some of them have told that explain clover leaf model in some of the papers. Some explanation is talk about the loops of tRNA. So tRNA is very important for you. I think it is one of board people's very favorite question. So please try to learn this, okay? tRNA has an anticodon loop. So students, tRNA looks something like this, right? Okay, I'm just going to okay, something like this it is looking. Okay, tRNA will look like this. Now they are telling that in one side it will have an anticodon loop. Which side is going to have anticodon loop? This side will have anticodon loop. Now what is anticodon loop meaning? What is anticodon loop meaning? Tell me in the chat box. Let's say this is your mRNA, okay? This is an mRNA that Girish took, okay? In Girish mRNA, I'm getting some, okay, let's say 3 genetic code I got AUG. I got AUG. After that, I got CUG. I got CUG. Now for this CUG, let's say one amino acid has to come and code, or I will use UUU because I know the amino acid, no? UUU. Now this amino acid is there, students. Now let's say this fellow, this tRNA is reading this, okay? He noticed that, okay, UUU is there. It will make anticodon. What will be anticodon of U? That is, who is going to code for U? So T is replaced by U, okay? So U is going to be coded by A. So A, automatically here who will come and attach? One amino acid will come and attach, which is phenylalanine. Okay, phenylalanine will come and attach here. Clear to all of you? Yes? Perfect? Okay? Yes, your NCRT, your tRNA diagram in NCRT it is given, but here you have to explain it, okay? So to make you understand, I am explaining this diagram for you. They will not ask you to draw the diagram, they will ask you to explain the diagram, okay? So tRNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor site. So this portion will be an amino acid acceptor site. Clear? This portion will be an amino acid acceptor site. That means, simple students, let's say I am reading it, okay? I am reading something like, you know, in olden days they used to write, no, like that paper I am reading, okay? Chota Bheem has given, okay? Dolakpur ke liye Chota Bheem has given one paper, I am reading the paper. Okay, this happened. So what will happen? At the same time I am reading, I read UUU. Suddenly in my head one idea will come, okay, phenylalanine, okay? One amino acid got coded. This is what is happening. Down in the anticodon loop will be reading, on the top amino acid accepting will be happening with the help of ribosomes. So that is what they have told. tRNAs are specific for each amino acid. That means tRNA is going to be specific. It's not going to be like, okay, some tRNA came, it code for something. No, because it has specific anticodon. Now, the tRNA that I drew can only code for A. That means it can code for UUU. It cannot code for anything else. So, they are very, very specific. The secondary structure of tRNA looks like a clover leaf model. In actual structure, the tRNA is a compact molecule which looks like an inverted L structure, okay? So 3D and 2D, they look different, okay? They look different. In 3D, they look like an inverted L structure. In 3D, they look like an inverted L structure, and in 2D, they look like a clover leaf. Clover leaf, all of you remember? Like in your phone, if you type lucky, okay, lucky, they will show you one leaf, no, that is your clover leaf. We call it as a lucky leaf. Okay, so it looks like that is what they are trying to say. Now look here students, this is what happens in your tRNA, no? So this is your ribosome unit, this is your larger ribosome, this is your smaller ribosome unit, this is your mRNA. Now what is happening? Can you see? One is coming and attaching, another fellow is waiting to come here, okay? As soon as this fellow comes, this fellow will go. So these what are these people? These are your tRNAs. They have just drawn it in a box form. Otherwise, it will become too confusing, no? So here you can see UAC will be done by this fellow. UAC will be done by this fellow. UAC this fellow. Now ACC another fellow. AAG another fellow. So what they are going to do? Let's say here one main person is there. Okay, this fellow will attach something. He'll get up and go. Then another fellow will be waiting to come. Like that, these people will continuously shift. Like how, you know, in chair, if let's say in a hospital queue, okay, in a hospital queue, there are a lot of people. Okay, lot of people. What will happen, students? One fellow gets up, another fellow shifts, and this fellow will come and sit here. Again, this fellow shifts. I will move this side. Right? Same thing is going to happen here. What is going to happen? Here, one tRNA will be there. He will come and wait here. He will attach the polypeptide chain. Next tRNA will come, attach, continue the chain. That's it. Okay, it is basically chain formation. No, entire polypeptide chain has to form. One tRNA will come with their anticodon loop, attach and go forward. That's it. Okay, it might be complicated for you when you're listening it for the first time, but do not worry. As I always told you, you can go and watch my lectures on molecular basis of inheritance. Recently only I did the lecture also. You can just type molecular basis of inheritance by Gopika ma'am. You will get lot of lectures of mine and you can listen to those classes. Okay, yes. What Saujanya put in the chat box, that is your clover leaf model. Okay, clover leaf model. Now, explain the salient features of genetic code. Now, in molecular basis of inheritance, I would request all of you from MBI, okay, molecular basis of inheritance, please learn transcription, okay, transcription, All the processes you have to study, students, because we can't predict which question might come. Okay, translation. These two you please study. I have not put here transcription and translation because I saw less question, but when you are studying for it, you please study it as a whole. Okay, you study it as a whole. Now, the salient features of genetic code. The codon is triplet. 61 codons code for amino acid and 3 do not code. That means these people are called as stop codons. Stop codons means what? Who is going to stop? Like when traffic police is there, he will stop. So he becomes the stop codon. Now, hence they are called as stop codons. Which are they? UAA, UAG and UGA. One codon codes for one amino acid. Hence it is unambiguous and specific. Some amino acids are coded by more than one codon. Hence the code is degenerate. Hence the code is degenerate. That means one amino acid itself. Let's say serine is there. Okay, I'm just writing. It is coded by UAG. Okay, it is coded, sorry. Let's say it is coded by UAC. It's coded by UAA. I'm just writing example, okay, but this is not the actual one. So like that means one amino acid can be coded by multiple codons because 64 codons are there. Okay, 64 codons are there. So multiple codons will be coding. The codon is read in the mRNA in a contiguous fashion. That means they're not going to read like UUU, comma, UAG, comma, no. UUU, UAG, UGG, like that. That means there is no full stop, punctuation, nothing is going to be present. The codon is nearly universal. That means from bacteria till humans. Okay, from bacteria till humans. All of them UUU will be coding for phenylalanine. This is one example that they have given, but what they are trying to say, from bacteria till, let's say 6.5 feet guy also, everyone is going to have, all the humans, be the smallest organism, be the largest organisms, all of them are going to have the same codon. That means the codes are nearly universal. Some exceptions are there, but then most of it is universal. Now, see here they have given the exceptions. Some exceptions of this rule have been found in mitochondrial codons. AUG has dual functions. Some codes have dual functions. AUG is a start codon plus it also codes for methionine. So look here, it codes for methionine and it also acts as an initiator or a start codon. Clear to all of you? This is there in your NCERT. Same thing I'm telling. Just have to learn those sentences of NCERT. Yes. So, the salient features of genetic code is what, students? Codes are triplet. 61 will code for amino acid, 3 will stop codons, UAA, UAG, UGA. And then you have the codons are degenerate. Codons are unambiguous and specific. Then one codon have multiple dual functions. It can also code for methionine at the same time. AUG can code for methionine at the same time. It can act as a start codon. Okay, all these points are very, very simple. At the same time, codons are universal. Be it bacteria, be it humans, UUU will code for phenylalanine. Yes, nonsense codon, correct. What is lac operon? This question also has been repeated so many times. Okay, very important. Multiple times it is repeated. They can ask you the diagram or they can ask you what is lac operon. Please explain lac operon. Okay, what is lac operon? How does it operate in the presence and absence of lactose? From the term itself, you understand lac operon. That means presence and absence of lactose will affect this model. What is first of all operon? What is first of all lac operon? Understand, students, every time do you people be active? No, sometimes you are active, sometimes you are not active. Sometimes you are very happy, sometimes you are very sad. We all have so many emotions. We also have some times of our day where we are extremely active. Sometimes we are like, no, I don't want to talk to anyone, right? Similar to that also here, what is going to happen? Sometimes the genes will switch off. Sometimes the genes will switch on. So these are like, okay, these are like luxury genes. Okay, today I don't want to work. Yeah, I will not work. Luxury people, right? They're like, okay, I don't want to work today. I will not work. Same thing here also. Genes are like, I don't want to work today. I don't feel like working. I will not work. But that feel comes from what? Because of presence or absence of something. That is, lactose is present. that means the gene is going to be switched on. If lactose is absent, the gene is going to be switched off. Okay, so look here students, this is the diagram given in your NCERT, similar diagram only, just different color I have taken. Look here, what is going to happen? I gene, okay, this fellow inducer is going to form repressor. This fellow's main job is to block or like stop the gene from getting switched on. So I gene will form repressor mRNA, repressor mRNA will form what? Protein. According to your central dogma, mRNA will form your protein, right? Protein will be formed. This fellow will run and go and sit on the operator. This O stands for operator. I've explained all these concepts on the channel, so now I'm just going to do an overview. Those who haven't seen it, please watch the video. Okay, so repressor mRNA will form repressor. That repressor will go and bind with the operator. Now what happens students? Enagutte illi who will be coming? RNA polymerase will be coming. RNA polymerase will be coming. RNA polymerase mastagi barta irtane. Oh yes, I can do transcription. But as it comes little more front, no? ಸ್ವಲ್ಪ ಮುಂದೆ ಬಂದ ಆಮೇಲೆ ನೋಡ್ತಾನೆ ಅಯ್ಯೋ ಆಪರೇಟರ್ ಮೇಲೆ ರಿಪ್ರೆಸರ್ ಕುಟ್ಕೊಂಡಿದ್ದಾನೆ, ಫುಲ್ ಬ್ಲಾಕ್ ಮಾಡಿದ್ದಾರೆ ರೋಡು, ನಾನು ಹೆಂಗೆ ಮುಂದೆ ಹೋಗೋದು? ದೇ ಕಾಂಟ್ ಗೋ. ಸೊ this RNA polymerase will not be able to do transcription. So that means here what is going to happen students? The gene is going to be switched off. Okay, gene will get switched off. Gene will get switched off. Artha aita? Okay, now second scenario, what is going to happen? Again, inducer will form repressor. Okay, again this will form repressor, protein also will be formed. This fellow will be running to go and bind with the operator, but it will not happen. Okay, it will not happen. Okay, that instead of that, what is going to happen? The inducer is going to be like your lactose. When lactose is present, inducer is going to be present. This inducer will go and attach to the repressor telling, I will not let you go. Okay, I am going to be with you. So inducer is going to be lactose. He will go and hold the repressor and he will tell, I will not let you go and bind with the operator. Now the RNA polymerase which is coming, he will happily come forward, and the Z, Y, A are structural genes. Z will release beta galactosidase, Y will release permease, A will release transacetylase. These also will be released, lac mRNA also will be formed, and finally transcription also will happen. Understood all of you? Lac operon concept is clear. When lactose is present, the gene will get switched on, that means transcription will happen. When lactose is absent, the gene will get switched off, that means transcription will not happen, because RNA polymerase cannot cross, because operator will be blocked by repressor. Okay, blocked by repressor. Moving forward, what are the various routes by which transmission of human immunodeficiency virus takes place? That is your HIV. Lot of questions on same pattern is seen about HIV. So look here students, sexual contact with an infected person. How does HIV spread? Always remember, HIV spreads mainly through blood and semen. Okay, semen. These are the main two ways the HIV virus is going to spread, right? HIV is going to spread. Now, in this, what is going to happen? First thing is sexual contact with an infected person through blood transfusion. Okay, by sharing infected needles, often among intravenous drug abuser. Have you seen those people who take drugs? They will inject it. Okay, they will do intravenous drug abuse. Now, what is going to happen? Let's say this fellow has AIDS. He has used the injection syringe and he has given to one of you. Now, what will happen? In that, there is blood, right? The syringe has come in contact with his blood. There is high chances that you will also get the HIV. Okay, now from mother to a child through the placenta. So these are the 4 methods by which your HIV can spread to you. That is mainly because of blood and semen. Okay, blood and semen. Students, it will not get spread through saliva, sweat and all of that, because very negligible amount of virus will be present and it cannot be spread. But higher amounts will be present in your blood and semen, so it will be spread through that. Okay, next one. How is cancerous cells different from normal cells? I think even cancer is one of your board's favorite question. They keep asking this question also. See, normal cells remain adhered to one another. That means, let's say we are all standing in a queue. Now next to me one person is there, next to me this side also one person is there. Suddenly I'm coming and standing here, I touch this person. I'm like, okay, I should stand properly, okay. That means what? I know that I have to stop, right? If let's say I'm a cell, I will stop my division when I come in contact with the next cell. That is called as contact inhibition, okay. So normal cells have something called as contact inhibition. Contact inhibition. That means when they come in contact with another cell, they will immediately stop the division and they will stay in that place, okay. So contact inhibition helps all the cells to be arranged properly. But students, cancer cells are ulta, okay. They will come in contact with a cell, but they will not stop the growth. They will keep growing, they will keep growing, okay. They will keep growing. They do not care for anyone. They will keep growing. So here you see they have a definite lifespan. Okay. Here also understand normal cells have programmed cell death. At one time the cell will die. Right. Now RBC also we tell no that your lifespan is 120 days. Now that means normal cells all have a program that is decided that you are going to die now, you will die. But this cancer cells do not have that. Okay. Cancer cells do not have programmed cell death. Now look here. Old cells will be replaced by new cells, which arises by cell division and differentiation. Cancer cells, whenever there is a breakdown of any regulatory mechanism, a cell develops the ability to undergo uncontrolled repeated division. Okay. So students, here what is the main point that you have to remember? Cancer cells, they have uncontrolled cell division. Okay, uncontrolled cell division. They have no programmed cell death. No programmed cell death. Next thing what they do, they have lost, okay, they have lost the ability, of contact inhibition. Okay, they have lost the ability of contact inhibition. And also you can write, in the case of malignant tumor, you can write metastasis. Okay, metastasis. Metastasis, that means they go from one place to another place, they will start growing, making tumor there. From there to here, making tumor. So they will keep continuing this. Okay. So these are the characters. Now, draw a neat labeled diagram of an antibody molecule. Now, you can stick to your NCERT diagram or you can stick to this diagram. I have drawn and shown you how you should be drawing it. Okay. Just draw two lines. Okay. Just draw a Y. And then just draw these two lines. Okay, even if you don't draw this also, it is fine. Just draw two Y like this. Okay, just draw one Y. Okay, one Y like this and just do this. This is your antibody. Simple as that. That's your antibody. You can easily draw it in the examination. Now look here students, what and all you are supposed to label there? You have to label antigen binding site. Okay, this is an antigen. This fellow will come and bind where? He'll come and bind here. That is called as antigen binding site. Okay, now other than antigen binding site, you have to label this as the heavy chain and light chain. Antibody is made up of 2 heavy chain and 2 light chain. Okay, 2 heavy chain and 2 light chain. See, I have written here H2L2. That means 2 heavy chain, 2 light chain. And what are they joined together by? These chains cannot be like, you know, oh, they will walk like this. No, they cannot do that. They have to be attached. Okay, they have to be attached by what? With the help of disulfide bonds. Okay, 2 identical antigen binding sites are going to be there. Here one antigen binding site, here one antigen binding site. So that means one antibody will have 2 antigen binding site. Artha aita ellargu? Yes? Clear? Any doubts? ಸ್ವಲ್ಪ answer ಮಾಡಿ students, ಸ್ವಲ್ಪ interactive ಆಗಿರಿ. Okay, be little interactive. What is this? Come on, answer. Likes are not at all nice, very bad likes. Okay, very, very disappointing likes. Tell me quickly. Okay, understood? Nothing is the antibody. What is antibody? They are basically protein. They are going to be like the bullets. Okay, like bullets, no? Same thing also here. Bullets will be there. They are going to catch the antigen. Antigen is what? The foreign particle. So this antigen will come. They will come and bind where? To the antigen binding site. Now what is this antibody made up of? 2 heavy chains, 2 light chains. They are joined together with disulfide bond. Can you see 2 S I have written? Disulfide bonds. Okay. How many disulfide bonds are there? All of you count in your NCERT and see 16 disulfide bonds are there. Artha aita? So 2 identical antigen binding sites will be there. Protein is there. This is how you can draw the diagram. Paratope, epitope, if you can remember, antigen binding site is called as paratope, and the portion where the antigen, I am the antigen, let's say my hand is going to bind here. This portion will be called the epitope. This portion will be called the paratope. So let's say, students, this is antibody, okay, where this chain is there, this is antibody. This is antigen. Okay, on the antibody, there is antigen binding site. That portion is called paratope. I am an antigen, I am a foreign particle, I am coming. Okay, I am coming. This portion, okay, which will bind with the paratope is called an epitope. Okay, which will bind with is called as an epitope. Now, this question also is being asked about your HIV virus. Okay, diagram generally they will not ask, but in case they ask a 5-mark question, you draw one Kuti diagram, not this complicated diagram. You can just draw this, okay, this, again you can draw from directly mRNA, then you can draw the nucleus, okay, nucleus like this, it's entering into the nucleus and ultimately forming something like this and then forming many viruses, okay, many viruses. That's this much you can draw, okay, in case it's asked you can draw like this. Now let me tell you what is given in the diagram. Okay, what is given in the diagram? So what is happening, students? The HIV virus is affecting our cells. Yes, ma'am. All of you should know that virus main capability is that this virus will release their genetic material inside the host. It will release the genetic material inside the host. So here also same thing happened. It released the genetic material inside the host. Now it has something called as reverse transcriptase. With the help of reverse transcriptase, viral RNA is introduced into the cell. Viral DNA is produced by reverse transcriptase. This can be asked as an MCQ. This can be asked as an MCQ. Who is going to help in the conversion of RNA to DNA? Okay, RNA to DNA. Who is going to help? Reverse transcriptase. Okay, reverse transcriptase. This fellow is going to do this job of converting RNA to DNA. So students, once they form the DNA, what is going to happen? They are going to use the viral DNA and they are going to incorporate into the host genome. Okay, our genome is going to incorporate into the host genome. New viral RNA is produced by infected cell, or you can directly write new viruses are produced. Okay, because once it enters into our host body, it will start manipulating, telling that, hey, you know what, you should not make yours, you should make ours. So it will tell, you make virus, you make new viral protein, you make new viruses, don't do whatever you're supposed to do, I will tell you what to do. So it will act like the boss, it will control our host genome. Okay, it will control our host genome. That is your virus. Next diagram based question that can come for you is describe the life cycle of malaria. So I have explained this multiple times for all of you, but still I am going to explain it again. What is going to happen, students? Remember, there is a digenetic host. That means 2 people is required for this Plasmodium vivax. First of all, malaria is caused by Plasmodium. What Plasmodium? Plasmodium vivax. Okay, Plasmodium vivax. Plasmodium vivax is going to be there, right? This fellow is going to need 2 hosts. One is going to be mosquito, female Anopheles mosquito. One is going to be the host. Host is who? Man. Okay, one man. So both are host. Okay, both are host. Now, here this fellow is going to act like what? A vector. But understand, one life cycle is going to happen inside their body. Okay. Now, here which life cycle is going to happen? I'm going to tell you here. Students, inside the mosquito, sexual phase of this Plasmodium vivax is going to happen, and in man, asexual phase will happen. Okay, asexual phase will happen. If they ask you an explanation for 5 marks, please write this, that sexual phase happens inside the mosquito, asexual phase happens inside man. Now let's see the life cycle. When they, so a mosquito, an infected mosquito will come and bite. This infected mosquito will have something called as sporozoites. That is why in biological classification in 11th standard you have studied sporozoans, okay. So these people come under sporozoans, okay, under protista. So here what is going to happen, students, this mosquito will bite. It will release sporozoites into the blood. From the blood it will first go to the liver, okay. First it will go where? It will go to the liver. In the liver, it will continuously multiplicate and it will burst open the liver cells. Once it bursts open the liver cells, it will go into the bloodstream again. From the bloodstream, it will get into the RBC. In the RBC also, it will multiply, break open the RBC. When they break open the RBC, there is a release of something called as a hemozoin. This hemozoin allows the symptoms of fever to occur. That is when a person gets to know that, oh my God, I am getting fever, I have to test myself. Why? Because hemozoin got released when the RBCs burst open, leading to causing fever. Now students, remember that when the RBCs, this entire multiplication is happening, no, male and female gamete will be formed. But in our body, we cannot do section. That means we cannot do fertilization in our body. So what will happen? The same mosquito can come, another mosquito can come. They will take the gametophytes in the blood mean. That means they'll take a blood, no? So in that the gametes will be there. They will do fertilization in the gut. Okay, they take place in the mosquitoes again. Intestine it will take place. That is here you can see. Here students, remember this is mosquito's gut. Okay, mosquito's gut. So in the mosquito's gut, what is going to happen? Fertilization will happen. Fertilization will happen. This fertilization will lead to the formation of sporozoites and it will be stored in the salivary gland, and then again the mosquito will continue this life cycle. So two main things that you have to remember here, is that the sporozoites, okay, fertilization, okay, let's not write sporozoites, I'll just write fertilization, okay, because fertilization is only happening in mosquito. Fertilization will happen in the gut of the mosquito, okay, female Anopheles mosquito, and storage of sporozoites will happen in the salivary gland, okay, in the salivary gland. Done? Clear? Yes, good job, okay. The sporozoites actually go to the liver because again they have to asexually reproduce, no? That is why they are going to the liver cells, okay? They are going to the liver cells. Now draw a neat labeled diagram of Miller's experiment, okay? Urey-Miller's, right? So here students, this is based on your chemical evolution, okay? I will get back to that, Girish, okay? I will definitely get a more valid reason about it and I will get back to you, okay? Now students, look here. Neat label diagram of Miller and Urey's experiment of chemical evolution. What did they do? In this setup, they took 4 gases, CH4, NH3, H2O and H2, okay, H2O and H2. They took all these gases inside a flask. In that flask, they, with the help of electrode, they gave spark discharge at 800 degrees Celsius. At 800 degrees Celsius. Because of the spark discharge, what happened, students? Condensation happened and it led to a liquid collection, water containing organic compounds. And when they checked the water, what is there exactly in this water? They were shocked to know that there were amino acids in this water. Okay, there were amino acids in this water. So from this, what did we understand? We understood by taking some inorganic substance, we can get some organic substances, okay, organic substances, correct? This was what Miller and Urey proved it, that is CH4, this students, these gases that they took, no, CH4, methane, NH3, ammonia, water vapor, all this were present in the early atmosphere. was present in the early atmosphere. So from the early atmospheres, you know, what was there in the early atmosphere, they took that and they gave electric discharge with the explanation, okay, that is Haldane and Operon and Haldane gave an explanation that there was a lightning. That lightning effect was given by the help of this electrodes. Okay, so because of this entire thing, because of this extreme heat and everything, the condensation happened and we got some organic substance. That organic substance was found out to be amino acid. Many other people also with the similar setup, by just changing these, they tried, okay, with changing these gases they tried, and some of them got fats, okay, some of them got pigments, some of them got sugars. All this also is given in your NCERT. Clear? They have given in your NCERT. Perfect. Okay. Moving forward, what is the difference between benign tumor and malignant tumor? Benign tumor means it's confined to one place. So I'm standing here only. I'm not going that side and this side. I'm standing here only. But if I'm malignant tumor, I will go here, I will go here, I will spread everywhere. Okay. So benign tumor are normally confined to one location where they are first developed. It takes time to develop. Oh, why is this? 1 second, students. Okay. Yes. So, are normally confined to one location where they are first developed. It takes time to develop and do not grow very rapidly, and most of the time they are not cancerous and they are not painful also, okay, and you can remove it with the help of doing some surgeries or operations, okay. Now malignant tumor on the other side, they are not like that. They are potential cancerous people. They will spread from one place to another place very quickly, okay. So they have potential to spread to other parts of the body. Their cells grow very rapidly and can spread in an alarming rate. The tumor spread into the organs that are near and rapidly split. Okay, all that is not needed, students. You can just write that they are cancerous and can be very, very dangerous. Okay, what and all did I tell you? Benign tumor is confined to one place. They will not move anywhere. They will just stay this place. Okay, they're like, hey, I'll be here only. And most of the time they're not cancerous and they're not painful. But on the other side, you have malignant tumor. These fellows will spread everywhere around the body and they are most of the time cancerous, very painful, very fatal, very dangerous. Okay, very dangerous. Artha aita? Okay. Now, what is adaptive radiation? Again, evolution question. This is a 2-mark question. Okay, this is a 2-mark question. Adaptive radiation. What is adaptive radiation? That is from one common ancestors. Okay, radiation has happened. Okay, you have radiated. Now some of you, your grandparents might be in the village, but all their, like all the, your, what do you tell, Tata Aji will be in like in Karnataka, some place or some village they will be, but your Chikkappa, Dodamma, okay, Chikkamma, all of you would have gone to different places. Maybe some of you are in Karnataka itself, some of you are in different places, like some of you are in Udupi, some of you are in Bangalore, some of you are in Hassan, some of you are in different places, no? That means adaptive radiation. You are all of a common ancestor, but you have all got radiated. But it does not have to be outside Karnataka. It can be inside Karnataka. Same thing here also, students. Adaptive radiation is the process of evolution where a single ancestral species, okay, give rise to variety of species, each adapted to a specific environment. That means you should understand from one point, okay, from one point radiation has happened. From one point radiation has happened. Okay. Now example what you will give? You will give Darwin's finches or your Australian marsupials. Okay. Darwin's finches in the Galapagos Island. What happened to Darwin's finches? They were all seed eating. Okay. They were all generally seed eating, but they got spread to different places in that island. One became insectivorous. Okay. One became vegetarian. Okay. That means started eating fruits and all of that, etc happened. Okay, etc happened. So from one point, if you radiate to different places, now after KCET, after boards, now all of you are classmates, no, all of you are in one college, all of you love going to college, but after some time you will not even have contact with each other because one of you might have gone to some other college, one of you would have taken different combination, some of you after second PU might go for commerce, or some of you might go for science, some of you might go for different courses. So you will become adaptive. That means from one single college you have all spread to different places. Okay, done. Yes, now define homologous and analogous organs. This is also very important question, students. Homologous organs with similar structures but different functions. Okay, similar structure but different functions. What are you going to think? My hand, what and all it has? Humerus, radius, ulna, carpals, metacarpals and phalanges. Now a cheetah also has the same thing. Whale also has the same thing. Okay, a bat also has the same thing. But one is, I am using it for holding something. Okay, I'm using it for holding something, writing, but cheetah is using it for running. Okay, whale is using it for swimming, bat is using it for flying. That means all of their structures are similar, but their functions are completely different. Okay, now students, how do you remember this? You remember like this: HDAC, HDTV ira roomali AC pakka irutte. Okay, HDTV in a room alli AC pakka irutte. In our homes and all it won't be there, but then necessarily a theatre. Just think of a theatre. HDTV idiyalva. So our place alla AC nu irutte. So HDAC. HD andre in students, H for homologous, D for divergent. Divergent. Ivru one point in the, from one point they are divergent. Okay, one is using it for running, one is using it for swimming, one is using it for catching. But on the other side, you have AC. AC is analogous. These people, organs with similar functions but different structures. Okay, AC means convergent. Convergent. That means they are different students. They are from different points, but they are all coming to one point. They are all coming to one point. This is called as your convergent. Okay, can you see the arrow mark different? From one point they radiated, from here they are coming together. Okay, example is wings of butterfly and birds. Maybe one is made up of extension of the forelimbs, one is made up of just of the exoskeleton, but both are used for flying. So why can it be different? Eyes of octopus and human, flippers of penguin and dolphin, all these comes under example. Potato, sweet potato, all of this also is an example. Okay, clear to all of you? Yes. Yes, now we'll learn biotechnology. Okay, biotechnology. Okay, describe the process of recombinant DNA technology. Okay, very important topic. You have to study this. Even my biotechnology video is available. All of you can go and check it out. Whichever chapter you want, you can go check and put my name. All the videos will be available. Okay, now you can check the recent videos. You don't have to check 9 months, 10 months back video. You can check the recent videos. Okay, yes. Now, isolation of the DNA. First thing, what? I want to do something. I want to make sure that this flower is blue in color. First, I will take my desired gene, right? I have to isolate the DNA because I'm going to play with the genome. I have to change the genetic material, otherwise no use. So, isolation of the DNA. The desired gene is extracted from the source organism. Now, cutting of DNA. I do not want the entire DNA. I just want a small portion of the DNA. So, I will cut it with the help of molecular scissors. Can you take a normal scissor and start cutting DNA? No, right? So, you will take molecular scissors to cut the DNA. What is molecular scissors? Other term? Tell me in the chat box. Molecular scissors? Yes, endonuclease, exonuclease is the endo which will cut in the middle, exo at the end. Okay? Nucleases also you can write. Usually we use endonuclease because we have to cut in the middle. Okay? Then formation of recombinant DNA. That is the gene of interest will be inserted into a vector. What is a vector, student? A vector is like a vehicle. Okay? Like a vehicle. That means what? It is going to carry my gene of interest. Very good, Chirag. Girish. Okay? It is going to carry my gene of interest. Okay? To the whatever desired place I want. No? So, vector is going to be the one who is going to carry that. Now, insertion into the host cell. Now, host cell will be there. Okay? Let's say this is my host cell. Okay, inside this host cell, I'm going to put this this is my vector, just an example. Okay, everything is just an example. I've added my desired gene here. Okay, now I'm going to insert this fellow inside. Okay, I'm going to insert this fellow inside. So here you go. I inserted this fellow inside with my desired gene. Okay, with my desired gene, it has now entered inside. Okay, understood, inside the host organ. Now selection of transformant cell, that is with the help of selectable markers like what? Ampicillin resistance, AMP, R, tetracycline resistance. I can check that which of this have actually worked. Some of this, what happens, no students, like when we do titration also. When we do titration, what happens? Suddenly dark pink will come, or suddenly pink will come and then it will vanish. That means what? We have not got near to the result. No, here also what happens? Some of them, this would have completely got transformed. That means I got a cell which has the desired gene plus the vector, everything is there. Some of the cell, the vector only would have entered. Some of the cell, the desired gene wouldn't have entered. Such kind of complication happens. To check those things, we have ampicillin resistant, tetracycline resistance, which are called as selectable markers. These people are going to tell us that who got transformed, who did not get transformed. Okay, then expression of the gene is expressed and the desired product is extracted and it is purified. That is downstreaming processing we'll be doing. Okay, those who haven't studied biotechnology, it's going to be difficult. Clear? Yes, I drank water. Okay, clear? Once again, let's go. Isolation of the DNA. I need to take the DNA, no, from where I need the desired gene. Then I will cut the DNA because I do not need the entire DNA. I just need a small portion of the DNA. Okay, let's say this is the DNA. Okay, in this DNA, I just want this portion. Okay, I just want this portion. Again, everything is a diagrammatic representation. Okay, I want this portion. So what I will do, I'll take molecular scissors and I will cut them. Okay, I will cut with the help of a molecular scissor. After cutting them, what will I do? I will make the recombinant DNA. That means with the help of a vector, I will put the desired gene into the vector and I'll get it. So the gene of interest, that is GOI also you can write. Okay, GOI, gene of interest, will be inserted into the vector using the DNA ligase. DNA ligase is a gluing enzyme, right? Now, insertion into the host, after doing this process, I will insert it into the host. Okay, this is the host. It could be an E. coli, it depends on what we want. Okay, after that, what you students, we will do with the help of selectable markers, I'll check. Okay, you got transformed. Okay, everything is proper. Then I will take it forward. They will express it. I will extract it, purify it. Okay, I'll extract it, purify it. Now, explain the steps involved in polymerase chain reaction. That is PCR. So, PCR is basically used for amplification of the DNA. Amplification of DNA. That means, students, from a small fragment, I can make millions of copies. Okay, from a small fragment, I can make thousands of copies, thousands or millions of copies. That is why we are using PCR, that is polymerase chain reaction. Okay, now for that there are 3 steps. I told you, how will you remember this? You tell bay, no, bay you tell, I guess. You tell, no, hey bay, but instead of bay you make it day. Okay, instead of bay you make it day. So D for denaturation, A for annealing, E for extension. So look here, D for denaturation, A for annealing, E for extension. Denaturation, DNA strands are separated by heating at 94 degrees Celsius to 98. Again 92 degrees Celsius also you can write. Okay, 92 degrees Celsius you can write approximately correctly if you want. Annealing, primers bind to the target sequence at 55 degree to 60. Again, it's a range. Okay. Now students, extension, DNA polymerase, that is Taq polymerase, synthesizes new DNA strands at 72 degrees Celsius. So please do understand one thing here. We are not removing the tube, adding the tube, removing, we are not doing that. We are just changing the temperature. I will do denaturation. That means, both the strands are there. I have to make sure I separate them. If I don't separate them, how will I make million copies of it? So first is denaturation, separating them. That will be at a higher temperature, 92 degrees Celsius. Okay. After that, what I will do, I'll take some oligonucleotides. Okay. If it's too difficult for you, just remember that we'll take some primers and we'll let this primers bind. Okay. So there is forward primer and reverse primer. Okay, one will be attached to the forward, one will be attached reverse. So these two primers will help us understand, okay, from here we have to continue. So primers will be added. After that, what will happen? Extension. We have to make the new strands. No, new strand formation will be taking place with the help of Taq polymerase, which is extracted from Thermus aquatis. So this is why we have taken this, because high temperature DNA polymerase cannot work. So we need another polymerase who can form the new template. For that, whom we will take? Yes, whom we'll take? We'll take Thermus aquatis. We'll extract Taq polymerase, which can withstand in extreme temperature. This is very thermostable. Okay, this is very thermostable. Very thermostable. So we will take this. Okay, this cycle is repeated multiple times until we amplify the DNA. Please students, if you haven't learnt the chapter, all this will be new to you. If you have learned the chapter, then all this will be easy to understand. Okay, otherwise biotechnology you cannot understand. So please watch a lecture if you want to understand. Now what is RNAi? That is RNA interference. Okay, RNA interference is a job, I told you last time also. RNA interference is a method of gene silencing where the double stranded RNA, this is important, can be asked as an MCQ. Okay, can be asked as an MCQ. RNA interference means what? I am interfering with something, no? Or we can also call it as RNA silencing. Okay, RNA interference is a method of gene silencing where the double-stranded RNA inhibits the expression of a specific mRNA. So there is a nematode that affects tobacco, Melidogyne incognita. This nematode was affecting tobacco for a long time. So we use this RNA interference to stop this nematode from affecting. How? mRNA to protein it will form. No, we did not let them make protein only. Then what happened? No problem at all. Okay, no problem at all. Right, so that we did. It is used in biotechnology to develop pest resistant plants. Okay, such as tobacco resistance to nematodes. Clear? Clear? I am not going into the details of it. If you start now by knowing something or by knowing nothing, by knowing nothing you cannot get full marks in board, students. You have to know something. Okay, you should at least know the concepts. Something you should know outline. Definitely you can practice from question papers, as I already made a video. You can practice from question papers. Whatever you know, you can definitely learn it faster. Whatever you don't know, if you're trying to by heart, it will take a lot of time. And I'm not a teacher who would love students to by heart. I do not believe in by hearting. Some boxes, tabular columns I understand, but otherwise by hearting is not my thing, because if you by heart, you will maybe forget the head or the tail and only write the middle portion and come. Okay, so you might not end up getting full marks. Okay, you might not end up getting full marks. Now, explain the process of production of genetically engineered insulin. Very, very important. Some dates are there, you have to remember. So, students, what happened? No, insulin structure I'll draw first for you to understand. So, this is A chain, okay. This is B chain. To keep these two chains together, we have disulfide bond. Okay, disulfide bond. Now, to keep this two chain again together, we have a C chain. Okay, I drew it wrong. We have a C chain. Okay, this is the structure of your insulin. Now I will label it. This is your A chain, this is your B chain, and this is your C chain. So when they were trying to make insulin, they were finding it too difficult to make insulin. Why? Because this insulin structure was not coming correct. Okay, because all of this was not coming in correct order. So what they did, there was a company called Eli Lilly. Eli Lilly in 1983. Okay, Eli Lilly in 1983. Okay, just give me a minute, students. Yes. Yes. So students, listen here. What happened? Eli Lilly company in 1983 figured out that actually we do not have to make the C chain, because the main problem that they were facing is to arrange this like this. Okay, this is insulin diagram. To arrange this, they were finding it so difficult. They were like, you know what, the C chain is only present when the insulin is in its pro state or inactive stage. So in the active stage, why do we need the C chain? So Eli Lilly was very smart. With the help of E. coli, okay, with the help of E. coli, they made insulin chain which had only the A and the B with the disulfide bonds. Okay, with the disulfide bonds. Clear to all of you? So this was how the isolation of insulin happened. They basically removed the C chain, telling that C chain is only present during the proactive stage. We do not need it in the active stage. So finally, Eli Lilly ended up forming humulin. Okay, humulin by biotechnology method. Students, first they tried it with the help of cattle, but cattle insulin was allergic to many people, so the only way was to make it like this. And finally this company called Eli Lilly, it is not some girl's name, Eli Lilly in 1983 found out humulin, where they used only the A chain and B chain. With the help of E. coli bacteria, they solved the issue. Okay, they solved the issue. So you don't have to read this. This explanation is more than enough. Okay, next is, how are transgenic animals produced? Mention any 3 application. Transgenic animals means what? Now let's say this is a normal gene of a, let's say this is a sheep. Okay, this is one gene. What I have done, I have taken this gene and I have manipulated it. That is, I have added a foreign gene into this. This kind of animals are called as transgenic animals. That means where their genes are manipulated. Okay, their genes are manipulated. Ma'am, why is this manipulation done? So that we can use these people for studying diseases. We cannot test it on us, so we will test it on these animals. Who are the most commonly used animals? Mouse, that is rat, mice. Rat is the most commonly used transgenic animal. Other than that, you have pigs, sheep, cow, all these are used also. Okay, so here you can see, you know, rabbits are used, pigs are used, sheep are used, cows are used, monkeys are used, fishes are used, but 95% of the transgenic animals are mice. Okay, mice. Now, why are they used? Because to study different diseases, to produce, okay, therapeutic proteins. That means to even find, you know, alpha-1 antitrypsin for emphysema. Okay, alpha-1 antitrypsin for emphysema. All this is in your NCERT. This is mainly given for emphysema, which is a disease caused because of continuous or chain smoking, we tell, no, because of continuous smoking. Organ transplants. Okay, organ transplants also we have tried, but it's not very successful. But we also use this to, you know, students, chemicals. Have you seen on this skincare brands, cream brands, they will write animal tested. That means they have tested it on animals. Animals did not have any allergy. Now you can use it. But nowadays, most of the cosmetics have been stopped or they have been banned from using these on animals. Okay. So organ transplants, production of therapeutic proteins, study of diseases, normal physiological understanding, then to test on toxic substances. All these transgenic animals are used. Now, what is transgenic animals? Animals whose genes have been manipulated or a foreign gene has been added into their genes. Okay. Here you can see animals that have their DNA manipulated to possess and express an foreign gene. Okay, to express a foreign gene. Okay, next, draw a neat label diagram of a simple stirred bioreactor. Students, this has been asked multiple times. In model question paper also is there, in previous year also it is there. Okay, you have to know this diagram. I just hope and pray they do not give it for drawing, because I don't know how you people will draw this. It looks complicated, but you at least make a simple diagram. Okay, you don't have to draw so much. At least try to draw this much. Okay, like this fan you draw. Okay, like this fan you draw. Like this fan you draw places, because it will take some time. Even if I draw now, it will take some time. Okay, bioreactor diagram is asked. Explanation I have not seen much. Only diagram is asked. Okay, so you should know what is it. Motor is there. Okay, foam breaker is there because continuous stirring will lead to forming the foam. Okay, so we should not make foam. Now why is bioreactor used? So that we can make it in large scale. Okay, large scale we can make it, right? Yeah, they will ask is what I believe, so please draw the diagram and learn. Okay, then flat bladed impeller, culture broth, this is your broth. Okay, now steam for sterilization, acid and base for pH control. Basically continuous stirring will happen, students, so that the oxygen is there and bubbles should not be formed, foam should not be formed. Bioreactors are basically used when you want to make something for large quantities, right? Like if I want to do something for commercial value, I cannot make it in a smaller scale, I have to make it in a larger scale. So that time I will use bioreactors. So diagram very important. Okay, diagram very important. Next one, draw a neat label diagram of PBr322 plasmid and explain the significance of selectable markers and restriction enzyme sites present here. This question also they can ask you the diagram or they can ask you to explain it. Okay, this is PBr32. What is PBr32? It is a cloning vector. Okay, it is a cloning vector. So students, why do we use this? Mainly because these people have selectable markers. See, they have ampicillin resistance and tetracycline resistance. They have an ORI site, that is origin of replication. If you people want biotechnology, please drop it in the comments if you want biotechnology. I will do an explanation of all the important concepts of biotechnology if you want, like a one-shot marathon. Okay, yes, but I don't know if it will be possible before your pre-boards. Please drop your biology pre-board dates. In case you wanted a day before that, I can try doing that. But for your PU examination, if you want biotechnology, you can drop it in the comments. I will do the chapter. Okay, yes, students. Okay, students, ampicillin resistance and tetracycline resistance is there because they both are selectable markers. Okay, then you have different types of restriction enzymes. See, all these are restriction enzymes. All these are restriction enzymes. I tried a lot to make a trick on this, but tricks are not coming at all because its letters are very scattered, so very difficult to make a trick. But again, we will still try to make a trick. Okay, so students, antibiotic resistance are there, both of this ampicillin resistant, tetracycline. These people are going to help for what? To understand if something is transformant or non-transformant cells. Okay, transformant and non-transformant. Okay, they are going to help in that. Then students, we have ROP, which will code for protein, that is which will code for your replication. Okay, this is going to help for replication. But all the other people are restriction sites. Okay, because restriction sites are going to be very specific. We will not have random in one vector. You will not see multiple restriction sites will not be there. Different restriction sites will be there. Okay. Evolution in neat English. Okay, I've done evolution so many times. I've done NCRT lines also. I have done RNA series also. Again, you people want evolution. Okay, last 3 chapters also is done. Just go and search. It is recently done. Again, doing that, it's going to be difficult, students. It's recently done. Go and check it out. Okay, on this channel, if you people want biotechnology, please tell me. Okay, 21st. Okay, fine. So this is here. See, E. coli cloning vectors BBR322 showing restriction sites. Hind3, EcoRI1, BAMH1, BAMH1, SAL1, PVU1, PST1, CAL1, and ORI and antibiotic resistant genes are there, which will code, and ROP is there, codes for the protein involved in replication of the plasmid. So this is a vector. You have to remember all of this because most of the time I have seen as diagrams. Okay, in your PU they have asked for diagrams. Explanation is very less because explanation is less and students will not even understand explanation. Okay, that is the problem. I mean, we will explain definitely. Okay, last question for the day. What are the use of genetically modified crops, or how are they different from the normal crops? Okay, look here students. These crops are made tolerant. Very important. These crops are made tolerant to abiotic stress. Be it cold, be it drought, be it, heat. These plants will be extremely, they'll be like, okay, No problem. Whatever be the condition, we will survive. Okay, these GMO means what? Genetically modified crops. We have modified the genes in them. Okay, we have changed, we have manipulated the genes in them so that they can give us better yield, they can give us better crop. Okay, and the post-harvest loss can be reduced. That is what is genetically modified crops. Genetically modified organisms also are there. Okay, here we are talking about organisms. That means you can write GMC, genetically modified crops. Reduced reliance on chemicals. Okay, chemical pesticides can be used less. Okay, help to reduce post-harvest loss, because students, after the harvest they will realize that, oh my god, this is infected, this is infected. So lot of loss will happen. Okay, that also can be avoided. Increased efficiency of mineral usage by the plants. Plants can take in more minerals, and it can not taken in such a way that the soil fertility will be completely gone. It will be taken in such a way that the soil fertility will be still retained. Okay, so look here. Increased efficiency of mineral usage by the plants. This will prevent exhaustion of fertility of the soil. Enhance nutritional value of food. Okay, we can increase the nutritional value of food, like how we did golden rice. All of you know, we added beta carotene. We added beta carotene, which was extracted from daffodil plants. We added this into Agrobacterium tumefacium, which is a plasmid, and we got golden rice. So people who had vitamin A deficiency, because this is a precursor, beta carotene is a precursor. Once it enters our body, it can form vitamin A. So this vitamin A deficiency, whoever had color blindness, okay, those who had that kind of issues could have consumed golden rice. Okay, biology 360 ಅಲ್ಲಿ ಮಾಡ್ತೀರಾ? Biology 360 ಅಲ್ಲಿ, if you want us to make the last 3 chapters, then Baswa sir has to take it alone, because there's no zoology there. So Baswa sir has to take it alone. I will tell sir, even you people can drop it in the comments. Okay, so vitamin A, that is vitamin A deficiency, can be reduced by having golden rice, because vitamin A enriched rice it is. Okay, I hope all of you understood all the questions. Okay, so students, those who are finding to understand difficulty in understanding the topic, let me tell you, go back to this playlist. Okay, go back to this playlist. It is PUC biology is given, that is only the biology part, but complete zoology is given. Molecular basis I have given. So whichever chapters you want, okay, whichever chapters or topics you want, it will be here. Otherwise you can also go on Vedantu NEET English. Okay, yes, longer lectures. Okay, if you're telling that, ma'am, I don't want shorter lectures, I want longer lectures, then go here, go to life. Okay, here, see, living world, this you people don't need. But then here, see, biotechnology unit, entire unit has been taken for 5 hours 16 minutes. You can take it as a one shot, like in 2x you can play and finish it off in 3 hours or 2 hours, you can finish it off. Principles is done, application is done together. Okay, this you can utilize. See here, genetics is done. This is for 8 hours. That is principles of inheritance and molecular basis of inheritance. Other than that, lot of videos are there, students. Playlist, if you go, see, you can see Tejas series is there. NCRT line by line is there. Okay, so many things are there. So many, whichever videos you want, everything is here. So you can use this channel also. If you like, you know, because pre-birth there's very less time. You might be like, ma'am, what to do? Don't take tension. You can follow this as well. Okay, I hope all of you loved the session. Please drop in the comments how you felt about the session. Do not fail to drop in the comments, because your 1 or 2 comments might let another student to open the video and see that, okay, this ma'am teaches nicely, because, you know, Girish has commented, Charan has commented, or Anika has commented, because of which another student might open it and be like, yes, I can open this video and watch. So you give the trust for another person, right? Now if you see normally some roast video or prank video, you see lot of people commenting on that, lot of people reacting on that, you will also feel like watching it. Similar to that. So you will only let us grow, and you will only help us reach to many students and capably help many students as well. So thank you so much, students. I hope all of you loved the session. So bega video na share ಮಾಡಿ, like ಮಾಡಿ, comment ಮಾಡಿ, friends ಜೊತೆ share ಮಾಡಿ, martogಬೇಡಿ. So don't be lazy, share it with your friends. Thank you so much for joining the session. See you all in next class.